This topic is about genetic engineering under the Earth and Life Science Quarter 2, Module 24. And let's try to see kung yung nakuha kong PowerPoint ay madadaanan din ang benefits of genetically modified organism, which is the Module 25 of the Earth and Life Science. So, genetic engineering. So, what is genetic engineering? Lesson objective. At the end of this lesson, you should be able to define genetic engineering, outline the process of genetic engineering involving some or all of the following: isolation, cutting, transformation, introduction of base, sequence, changes, and expression. Know the three applications: one plant, one animal, and one microorganisms. Genetic engineering. Let's see. May lumabas ng definition. Dito tayo. So, the simple addition, deletion, or manipulation of a single trait in an organism to create a desired change. So, when we, di ba sa genetic is yung ang pag-aaral siya ng paglilipat ng traits from the parents to the offspring. Now, when we say genes, it contains the DNA. It has the characteristic of a certain organism, or it possesses the characteristics of a certain organism. When we say genetic engineering, meron tayong ginagawa doon sa genes na pagbabago. So, the process is simple addition, deletion, or manipulation of a single trait in an organism to create a desired change. Kung gusto mong mag-iba yung, let's say sa halaman, kung gusto mong may kakaiba dun sa kulay ng bunga, may gagawin kang bagay para lumabas doon sa bunga ng halaman. So, sa animals, ganun din. Kung gusto mong may ibang kulay ng balat or balahibo, may ilalagay ka sa genes niya para mabago yung kulay nung balat or balahibo. Pwede din sa mga, sa mga halaman, sa mga gulay. Let's say, pabibilisin naman natin yung pagtubo nung gulay or pagbunga, genetic engineering din yon. Now, what you need to know about genetic engineering? So, first, manipulation and alteration of genes. Minamanipulate natin, dinadagdagan, binabawasan, binabago. Process involving isolation, transformation, and expression. Next, 3 applications: one plant, one animal, one microorganism. Artificially copying. When we say genetic engineering, it is artificially a way of artificial copying of a piece of DNA from one organism and joining this copy of DNA into the DNA of another organism. Parang pahihiramin kita ng DNA kasi gusto ko yung traits ko ay nasa iyo or vice versa. So, pangungopya ng DNA from one organism transferred to another organism. Now, what is the purpose of genetic engineering? It allows genes from one organism to be inserted into a cell of a different organisms of a different species. And then, given our examples, human genes can be inserted into a bacterium. Human genes can be inserted into cells from other animals. And then, bacterium genes can be inserted into plant cells. So, those are examples nung tinutukoy natin na paano yung insertion sa cell from one organism to another. Ito yung mga yon. Next, it is also means that the DNA from different organisms can be combined. Kanina, nag-insert ka, nagdagdag. Ngayon, pwede mo silang pansamahin naman. Bacteria can be engineered to produce human proteins. Human genes can be inserted into other animals. So, this is the overview. Altered DNA is called recombinant DNA. When we say recombinant DNA, the DNA is joined to other unrelated DNA in the organism. Hindi sila related when it comes to characteristics of traits, pero pinagsama mo. This is called gene splicing. Next. When we have this gene splicing, I will check myself dun sa pronunciation ito. Splicing. Let me check it. Tiny segments of a gene are taken out and replaced by different genes. May aalisin ka, tas may ipapalit ka. So, gene splicing. Next. So, learning check. Okay. Pasagot nito. Sa inyong reflection. First part. Papaisa. Sa reflection part natin, what is genetic engineering? What is the purpose of genetic re-engineering? And give 3 examples of genetic engineering. Now, what is the difference between altered and recombinant DNA? And then lastly, yung ating binigay, what is meant by gene splicing? Next. Transgenic organisms. Organisms altered by genetic engineering, genetic material changed by other than random natural breeding, and then gene transfer or moving a gene from one organism to another. So, yung recombinant DNA kanina, under siya ng module 23. Module 24. Dalawa yung module na tinatry kong i-cover with this discussion. The module 24 and the module 25. Module 24 is genetic engineering. Module 25 is benefits of genetically modified organisms. Now, we are turning into this topic, transgenic organisms again. Organisms altered by genetic engineering, genetic material changed by other than random natural breeding, and gene transfer. Now, what transgenic means? Trans means crossing from one place to another. The genic bit means genes. So it means that bits of genes from different living things have been bolted together and spliced into another organisms to make a new one, which does something which the scientists want it to do. Next. Examples of transgenic organisms, GMO, genetically modified organisms, and GEO, genetically enhanced organism. For example, plants that resist a particular type of weed killer, and sheep which make some special substance in its milk. Kita nyo yung strawberry, may kulay blue. Genetically modified organism, wherein ang usual niya color is red and the reddish pink, mga ganun. Next, learning check. Mag-ano kayo sa reflection notes nyo ha, vocabulary. What the word transgenic means, what is a transgenic organism, and give example of transgenic organism. 5 stages involved in genetic engineering. First one is isolation. The second one is cutting. The third one is ligation and insertion. The fourth is transformation. And the last one is expression. So in isolation of a specific gene from a donor, example human, the cells broken open. This is the donor DNA. Genetic probe added. This is the genetic probe. And the reveals position of the gene of interest. Yun. Ang position of gene of interest. Yung area na ito. Let's see. Itong area na ito. Ang gene of interest. Isolation of a plasmid from a bacterial cell. So, eto, pakita natin ng konti. Wait lang, ha? Wala. Wala, wait lang. Wala. Technical error ni ma'am. while doing the ay, sorry. I want the pointers kasi. Nakita niyo yung pointers? Ayun na. I want the pointers. Ano, wala. Wag na akong gumamit ng pointers. Turo na lang natin. So, yung bacterial cell, this is the example of bacterial cell. Tapos, yung plasmid, ayun, may bigger picture siya dun sa ilalim. 1 micrometer. I-isolate from a bacterial cell. So, ilalabas. Next, cutting. Restriction enzymes act as molecular scissors and cut DNA at specific sites called restriction sites. So, meron lang specific sites sa DNA ang ikakat mo. Yun ang area na yon. Ayan, ikot-ikot pa siya. Yan ang area na ikakat mo dun sa DNA. The green part is the restriction site. Yung gunting, yung restriction enzymes. Then, cutting. Kita, the donor DNA, the restriction site. And these are the restriction enzymes. They are cute. Next. So, remember dun sa ating discussion dun sa genetics, 'di ba? Meron tayong pinipair na nitrogenous bases. Cytosine, guanine, thymine, adenine, yan yon. So, dun may kinakat lang tayo, may pagdudugtungin tayo, yung thymine, adenine, idudugtong mo dun sa dulo, adenine, thymine. Yan. And then, the DNA ligase, yun. Ligation or the rejoining the cut fragments of DNA and forming artificial recombinant molecules. So, kita nyo na recombinant kasi magkaiba naman sila ng kulay. So, hindi talaga sila yung iisang DNA. Pinagsama lang silang DNA. DNA of a certain organism and another DNA of a certain organism. Then, joined together by ligation. So, DNA ligase. Next. So, this is the picture or diagram of DNA ligation and insertion. So, yung foreign DNA, may restriction enzyme, papasok dun sa hybrid plasmid. And then, R plasmid, restriction enzyme, papasok dun. So, ligation, insertion, magkaiba sila ng structure kasi magkaiba silang DNA, pero pagsasamahin natin sila doon sa hybrid plasmid. Transformation, so bacterial chromosome, bacterial cell, recombinant DNA, yun na yung clone genes o yung kinopya natin na genes. Or ginawa natin na genes. So, recombinant DNA introduced into bacterial cell. Expression, bacterial cell reproduces by binary fission. Nag-cut tayo, di ba? Binary fission. So, yun. From clone gene, ayan, pwede pa siyang i-clone mo pa ulit ng madami. Bacterial cell produces the polypeptide coded for by the donor DNA. So, summary of steps. Cut with restriction enzymes. Dalawang donor DNA and then ligase bonds, sticky ends together. And then the recombinant DNA will be formed. So, yun yung summary natin ng steps. So, screenshot. Isama nyo yung mukha nyo sa screenshot para makita kung nanood kayo ng video lesson ni ma'am. Next, expression is getting the organisms with the recombinant DNA to produce the desired protein. When the protein is produced in large amount, it is isolated and purified. Again, doon naman sa expression, meron ka kasing gusto nga makita kaya kagagawa ng recombinant DNA. Kaya ka nag-undergo ng genetic engineering kasi may gusto kang bagay na makita doon sa iyong ginagalaw na organism. Next. Learning check. Sulat nyo ulit sa nyo. What are the stages involved in GE? Outline what happens in the stage isolation. And what enzymes is used to cut the DNA plus? Can you explain what happens in transformation? Putulin ko kasi mahaba na to. Ganyan na lang kayo sa 5. What is meant by gene expression? Putulin ko doon sa una, hanggang dito lang. Tapos, open ko na lang ulit ito bukas para naman makagawa muna kayo ng output for the genetic engineering. And may additional pa akong voice recorded lesson about this. Yung mismo namang nasa module natin. Module 24 and 25. Thank you and God bless.